Hello everyone, this is Tejas Saudari. I hope you have seen the part first of the translation in prokaryotes video, which was having certain animations and narrations which were explaining the first step of the translation process, that is initiation of the translation. Now, we are moving to the next important process, that is elongation, and after that termination. So, in this video, you can observe, you can listen carefully and understand what happens in elongation and termination process. So basically, in prokaryotic translation, we have to understand these three processes very carefully. So let's start with it.
After an initiation complex is formed, elongation occurs. During elongation, the ribosome synthesizes proteins by linking together amino acids. 3 steps are needed to add each amino acid. A charged tRNA is placed in the A site of the ribosome. A peptide bond is formed, and the tRNA with the growing protein chain is moved from the A site to the P site. In prokaryotes, this requires 2 translation elongation factors, EFTU and EFG. A third elongation factor, EFTS, is needed to recycle EFTU. Elongation is very similar in eukaryotes, and only the prokaryotic process will be described here. When the triplet anticodon of the correct tRNA is base paired with the mRNA in the ribosome A site, the base pairing is strengthened by additional hydrogen bonding with the ribosome. Incorrectly bound tRNAs do not base pair as strongly with the ribosome and are released before they can be added to the growing polypeptide chain. Correctly base paired tRNAs have additional hydrogen bonds that help the tRNA remain in the A site during accommodation, the process whereby the tRNA rotates into position for peptide bond formation. First, a charged tRNA is bound by EFTU and is brought to the ribosome A site as an EFTU GTP amino acyl tRNA complex. The tRNA anticodon binds the mRNA codon, and EFTU hydrolyzes its GTP, and the resulting EFTU GDP is released from the tRNA. Next, a peptide bond is formed between the amino acids. This reaction transfers the amino acid from the tRNA in the P site onto the amino acyl tRNA in the A site. To prepare for addition of the next amino acid, the ribosome undergoes translocation, a process in which the ribosome moves along the mRNA to the next codon. Translocation is facilitated by elongation factor EFG. The tRNA with the growing amino acid chain attached moves from the A site into the P site, and the spent tRNA in the P site moves into the E site. Note that elongation factors EFTU and EFG both end up as complexes with GDP after one round of elongation. Both factors must exchange their GDP for GTP before they can be used again. For EFTU, this exchange is facilitated by elongation factor EFTS. For EFG, the GDP simply falls off and is quickly replaced by a GTP. The process continues. At each codon, the corresponding amino acyl tRNA binds in the A site. The growing peptide chain attaches to the amino acid on the tRNA in the A site, and the ribosome translocates, moving the growing protein chain and tRNA into the P site, and freeing up the A site for another round of amino acid addition. Termination of translation is engaged when a stop codon on the mRNA enters the A site of the ribosome and is performed by release factors that recognize the stop codon. The mechanism of termination is similar in prokaryotes and eukaryotes and involves two kinds of release factors called Class I release factors and Class II release factors. Class I release factors enter the ribosome A site and bind the stop codon using a peptide anticodon. Prokaryotes have two Class I release factors, RF1 and RF2. Eukaryotes have one Class I release factor, ERF1. The Class II release factor, in complex with GDP, also binds the large subunit. In prokaryotes, RF3 is the Class II release factor. In eukaryotes, ERF3 is the Class II release factor. After the Class I and Class II release factors are both bound, a glycine-glycine-glutamine, or GGQ, motif on the Class I release factor triggers hydrolysis of the protein from the tRNA in the P site. Release of the protein from the ribosome triggers the recycling of the release factors. First, GTP is exchanged for GDP on the Class II release factor. This causes the Class II release factor to push the Class I release factor off the ribosome. The release of the Class I release factor allows RF3 to interact with the factor binding center, which stimulates RF3's GTPase activity. GTP is then hydrolyzed on the Class II release factor. The Class II release factor GDP complex then releases from the used ribosome. At this point, the ribosome still contains two deacetylated tRNAs and is also still bound to the mRNA. The process of cleaning out the ribosome and separating the subunits is called ribosome recycling. Although not as well understood in eukaryotes, in prokaryotes this process involves a ribosome recycling factor plus initiation factor IF3 and elongation factor EFG. The ribosome recycling factor, RRF, binds to the A site of the used ribosome and recruits EFG to the complex. The RRF acts like an A site tRNA during the translocation process. EFG can thus push on the RRF in the same way as it would on a tRNA. This moves the RRF into the P site and releases the two spent tRNAs. Next, both the RRF and EFG uncouple from the ribosome. The mRNA is released, possibly with the aid of IF3. IF3 binds the small subunit to prepare it for another round of protein synthesis.